Thank you very much, Shirai Sensei, for your kind introduction. And I'm going to talk today about the subject here, antibody-antigen modeling in a high throughput manner. And before I get started, I will reintroduce my background, although it's repeat some of the things that Shirai Sensei said. So I did study chemistry originally, and when I was just a first-year chemist student, I wanted to do experiments, and that didn't happen. I got ended up in a computational lab, and my professor was Richard Friesner, and he actually started a company called Schrödinger, and I ended up working for that company for 5 years. After that, I came to Japan and worked at the Institute for Protein Research, and I stayed at Osaka University for a kind of long time, longer than I planned originally. I planned to just refresh in Japan for a few years, but somehow the time flew by. And I also worked at Kyoto University for 2 years, and even I started my own company, maybe try to copy my Professor Friesner. And I will mention about this company a little bit in my talk. Then I returned to Osaka University from Kyoto University. I'm still at Osaka University at the Research Institute for Microbial Diseases. Okay. So my talk has a little different parts, and I start out not talking so much about science, but just about my personal experience relating to the topic today, which is antibodies. So try to answer the question, why I got interested in antibodies. And this is my group, research group now at Osaka University. And quite a few people in this group are working on antibodies, plus me. So that's a spell mistake. Should say half, about half. About half of us are working on antibodies. So you can appreciate that my lab is quite dedicated to antibody-related research. And you might think that I was interested in antibodies for a long time, but actually it's not the case. I got interested in antibodies completely by chance. And that was in 2013. So our group got invited to participate in antibody modeling assessment. So it's a kind of contest. And the purpose of this contest was to assess the state of the art in antibody 3D modeling. And this contest is organized like a CASP or Capri, if you're familiar with these contests, in which the organizers provide sequences of antibodies. And then the participants are supposed to use those sequences to build models. And then we send our models back to the organizers. And then the organizers assess those models. So it's a blind contest. We don't know the answer when we're doing our work. And there were, it looks like 7 participants, and most of them are actually companies, like Cellaris, CCG, Schrödinger, the company I used to work for, Macromoltec, and Astellas Pharma, which was the team we were on. And the 2 academic groups, Jeff Gray's group at Johns Hopkins, and the Pigs group in Rome. And the Jeff Gray is the former postdoc of David Baker, and so he's using Rosetta. So this is a Rosetta group. Okay. And the organizers were both company people and academics. So Johnson & Johnson and Pfizer were supporting this, but also some people at Scripps Research. And our team was headed by Haruki Nakamura, my former boss at Institute of Protein Research, and Hiroki Shirai at Astellas Pharma was the one who was actually invited. And so he recruited Nakamura-sensei, and I think Nakamura-sensei recruited me, and we made a team. And it's nice that Shirai-san was on the team because he has experience with antibody modeling. Maybe Nakamura-sensei has some experience, but I definitely had zero experience in antibody modeling. So our strategy was actually to make a redundant approach. So actually my team and the Nakamura team would independently predict models for each. And then we would choose the best models by looking at them and discussing them in these very intense meetings. And I didn't have a method to predict antibodies, so I had a 1 month and we wrote some software to predict antibodies. And our approach is fast approach. So basically, we just try to take pieces of structure and assemble them together and score them. And Nakamura-sensei took the very classic approach of doing molecular dynamics for a very long time and tried to sample the antibody loops as much as possible. And to get to the point, actually, our predictions as a team were overall the best. And so this is showing the errors. So lower is better here. And you can see this is the average error of the CDR3 loops. So something like a little less than 2.5 angstroms. And this was a shock to everyone, especially us. We didn't expect that we could do as well as Schrödinger or Rosetta. And so this, of course, got me interested in antibodies because, you know, it's not like I win contests every day. Now, one of the things that we found out, this is maybe a little bit hard to see, but anyway, is that if we had not done two methods, if we had just done the fast method, we would not have performed worse, about the same. So this is a very interesting lesson that a lot of molecular dynamics was done on each model. And also my group did the quick way, which is just using known templates to get the structure. And it turns out that the quick way was overall the same level of error, the same level of accuracy as the slow way. So this is an important lesson that sometimes you doing a complicated algorithm, it's not always better. And that's a problem in bioinformatics. Sometimes it's better, sometimes it's not better. So we discovered that in this particular case, the payoff for doing molecular dynamics and the cost of doing molecular dynamics kind of canceled themselves out. And so we pursued this quick way and we made a software called Kotai Antibody Builder. And we showed that, yeah, if we ran Kotai Antibody Builder, let's see, that is with, if we didn't do any refinement, we didn't do as well. These are the errors again. So errors were higher, and the green here is the, how we did in the contest. But if we just did a 1 hour of refinement, we could reach the same level of error. That's kind of interesting. And it definitely influenced my approach to doing antibody modeling. And it made me ask my assistant professor at the time, Kazuo Yamashita, should we make a company? Because if we beat Schrödinger, hey, why not? And he appropriately said, we? So he had to make the company. He's still the CEO of this Kotai Biotechnologies, and I guess it's pretty tough to be a CEO of a company. It's easy for me to say, let's make a company when I don't have to do anything. But anyway, I'm cheering for him. Okay, so that's the end of part one. That's my story of how I got interested in antibodies. So I'll just try to summarize. So the contest, assessment of antibody modeling assessment 2 provided an important lesson for me. And although I had no experience in antibody modeling, we were able to make an important breakthrough, which is to make something that was at least as accurate as anything out there in a blind test in a very efficient, speedy way. So why did we do so well? I think it's possible that because we were not experts, so we didn't know anything. So we didn't know what we were doing was stupid or it was naive. And so we were lucky. It happened to work. It just happened to work. And this is important to keep this lesson in mind for the students who are listening. So you will probably be assigned a project and try something, and it typically easy to get attached to that project. It's easy to get attached to your way of attacking the project. But some projects just don't work, or your way of attacking the project doesn't work. You shouldn't care so much. You can try something different. So my rule of thumb is now, if I work on something for 5 years and I didn't make progress, maybe time to try something new. And you shouldn't feel shame about that, because accidents happen all the time. And I believe that most scientific discovery is through accident. So let those accidents work for you and not against you. Okay. So that's the end of the non-science part of this talk. The rest is going to be boring science stuff, so I apologize, but I will try to go slow and don't make it too boring. So the next part is not really about my research. It's just general information about antibodies. So I hope that you will find some of this useful. So the first question is, where do antibodies come from? How does our body make antibodies? And the best answer I could make is, it's very simple. You have an antigen, you put it in an animal, and you wait about a week, and you get B cells that react to that antigen. Those B cells have B cell receptors, which are generally unique to each cell. And the chances are that the most populated B cells a week after you immunize an animal will bind to the antigen, and the antibodies are just the soluble portion of the B cell receptor. So this is not a physics explanation for how nature makes antibodies, but this is how we make antibodies, is that we expose an animal, usually a mouse or a rabbit, to an antigen, and the antibodies just come almost every time. So it always works. So each antibody is, well, it depends on the antibody, but typically the IgG antibodies that we work with are bivalent. They have 2 arms, which are identical, and each arm contains one of these Fabs or FABs, and that is the contains the receptor here that binds to the antigen, and this Fab is composed of 2 chains, a heavy chain and a light chain. And each of these chains has 3 important loops that we call complementarity determining regions, or CDRs. So I'm going to say CDRs, and I just want you to remember this looks like a beach sandals image with your toes sticking out. Those are the CDRs, and that's the important part of the antibody. That's what makes it different from other antibodies. So CDRs give antibodies their unique antigen binding characteristics. So let's look at a real antibody, not a PowerPoint antibody. Now we're looking down, we're looking straight at the CDRs. And by the way, this group of CDRs, all 6 of them together, it has a jargony name here, paratope. So we call it paratope. So I will say paratope sometimes, and I just want you to imagine the surface of the antibody that contains these CDRs. Okay. Okay. And these CDR sequences are generated randomly inside your body, inside the B cells, like this. So each B cell makes a certain combination of CDRs, and you have so many B cells in your body that you have so many different antibodies. This is continuous. This is happening right now in all of our bodies. If we are healthy, we are making new B cells with new CDRs. We never stop. And the number of possibilities in this CDR space is astronomical. It's so big, you can't really grasp it. But one thing that you can imagine is it's roughly equal to the number of grains of sand on Earth. That's roughly the number of different antibodies that humans can make. So big number, 10 to the 19 or something like that. Okay, so this slide just shows what I showed you before, but it has some, it's a movie. So when we look down at the paratope, we see 2, 4, 6 CDRs, and they make a continuous surface that has unique chemistry and it has unique shape. And it is the chemistry and the shape that determines the unique antigen and unique binding site on that antigen that each antibody is associated with. Okay? Okay, so we covered the paratope. Now let's talk about the epitope. The epitope is on the antigen. So where the antibody binds the antigen is called the epitope. Now I said that the paratope is on the CDRs. You might think that the epitope is some special place on the antigen, but no. Unfortunately, the epitope is anywhere on the antigen that the antibody can bind. So this is an example. Here, what I show you is hemagglutinin from influenza in green, and this is just another view of it. And what you see in magenta and yellow are all the known antibodies that bind to hemagglutinin. And what I want you to appreciate is that almost every available surface patch on hemagglutinin is targeted by some antibody. The only reason you don't see them here is because I didn't show them here. This is a trimeric symmetric molecule. So I just showed one of the hemagglutinin here being targeted. So it's the same with other viruses, HIV, COVID-19, spike protein. The whole entire surface of their antigen can be targeted by different antibodies. That's why you need multiple antibodies in your body. Okay, so this summarizes in a kind of graph what I just said. The paratopes are restricted to the CDRs. You see here, 3 peaks, these red bars indicating the, I don't know where that came from, but anyway, the 3 peaks indicating the frequency of being at a paratope, CDR1, CDR2, CDR3, CDR1, CDR2, CDR3. This is the heavy chain, this is the light chain. However, when we look at the same statistics for epitopes, again, on hemagglutinin, it's anywhere on the surface. Anywhere on the surface can be a epitope, okay? Okay. So remember I said that the body is making these B cells right now throughout your life. And it makes the B cell receptors randomly. So you might think because we're doing this and because they're so important, you might think that it's an efficient system, that somehow our bodies have figured out how to make good antibodies and not make bad antibodies. And this is completely not the way it works. The body is so inefficient that 95% or more of the B cells that it makes randomly get killed inside your bone marrow, inside your bones, because they fail a very important test which is the B cells should not bind to your own proteins. You shouldn't bind to human proteins if you're making antibodies. That's because they will attack you and you'll have autoimmune disease like rheumatoid arthritis or lupus or any of another vasculitis, lots of diseases that are caused by antibodies targeting your own tissues. So the body is very, very strict about removing potentially harmful antibodies, and that results in at least 95% of them being killed before they ever leave the niche where they are first developing inside your bones, okay?